Dear students, in this lecture we will be discussing sexual reproduction in plants. Students' learning outcomes. First SLO is describe salient features in the life cycle of gymnosperms. Explain the structure of flower, pollination and its types. Describe alternation of generation in plants. Explain process of sexual reproduction in angiosperms. Describe seed dormancy. Explain epigeal and hypogeal germination of seeds. Explain process of vernalization. Let's start. First SLO is salient features in the life cycle of gymnosperms. You already know that what are gymnosperms. Gymnosperm is the group of plants which have naked seeds and they are the evergreen plants. So their life cycle involves alternation of generation. You all know that what is alternation of generation. Alternation of generation is actually plants having two types of generations like sporophyte generation and gametophyte generation. Same is with the gymnosperms that they have a dominant sporophyte generation and a reduced gametophyte generation. Sporophyte is dominant because the mature plant is sporophyte and it gives rise to gametophyte, and gametophyte is dependent on sporophyte for food and other nutrients. That's why it is reduced and sporophyte is dominant. So what is alternation of generation in this case? Sporophyte generation gives rise to gametophyte generation and gametophyte generation give rise to sporophyte generation. So there is alternation of generation in the life cycle of gymnosperms. All the gymnosperms are heterosporous. What does that mean? Hetero means different, spores means having the ability to produce different spores. So gymnosperms also produce two different spores, namely megaspore and microspore. Megaspore will give rise to female gametophyte and microspore will give rise to male gametophyte. So what happens? The functional megaspore develops into female gametophyte, that is archegonium, that produce the ovum. So here how the sporophyte is giving rise to gametophytes. See, this was the dominant sporophyte. It has the ability to produce two types of spores, megaspore and microspores. In case of female, the megaspore is going to give rise to female gametophyte, and female gametophyte is that part of the plant which has the ability to produce female gametes. That's why it is known as gametophyte. On the other hand, microspore develops into male gametophyte, that is pollen grain, and the pollen grain that consists of two types of nuclei, tube nucleus and generative nucleus. This generative nucleus produces sperm, so that's why the pollen grain is the male gametophyte. Microspores, how the life cycle continues. Microspores are released into the air as pollens which land on the ovule cone, and fertilization usually occurs over a year after pollination. So this was a brief summary. Let's try to understand the life cycle or the salient features of life cycle of gymnosperm with the help of a diagram. Here you go. In this diagram, it represents the life cycle of a gymnosperm. Here you can see, from here we will start, this whole plant, the dominant plant, the mature plant, it is known as sporophyte generation. This sporophyte generation contains two types of cones, pollen cone and ovule cone. Pollen cone is the male cone. Here you can see the sign, and ovulate cone or ovule cone is the female cone. This pollen cone consists of structures scales which are known as microsporangium. These microsporangium have cells which undergo meiosis. Here you will see that till this part it was diploid generation, having the complete set of chromosomes. From here the microsporangium, the cells of the microsporangium which are known as microsporangiocytes, they undergo meiosis, and when meiosis takes place, you better know that the resulting cells of meiosis are haploid cells. So they produce haploid pollens. This pollen, a single pollen, here you can see this is known as male gametophyte, and it has wings and it has two nuclei, generative nuclei and tube nuclei. So the wings actually help in the male gametophyte to fly and land on the female gametophyte. Their fertilization can take place. We will come back to this point. Now we will discuss the female part, what happens in the female cone. Same is as like the male cone. Here there are also scales which are also known as macrosporangium. The cells of macrosporangium undergo meiosis and they produce megaspores. The pollen grains were microspores, and microspores, they produce pollens. Here megaspore gives rise to female gametophyte, which is archegonium. This archegonium has the ability to produce the eggs. So archegonium is the female part and it has the egg. Pollen is the male part, male gametophyte, and it has sperm. So when it lands in the archegonium, their pollen tube helps to form a pathway, and then the generative nucleus produces the sperm, and the sperm can combine with the egg cell. But fertilization, this was pollination. When the pollen fall on the archegonium, this is pollination. But a unique feature of gymnosperms is that here fertilization does not necessarily take place soon after pollination. It can take many time. It can take even a year to take fertilization. Fertilization is pollination is followed by fertilization, which can take place even in a year. So when fertilization is successful, there is the seed has the embryo, and this seed, when it falls on the ground, it has the ability to produce a sporophyte plant, new sporophyte plant. So this is alternation of generation. There the sporophyte generation give rise to gametophyte generation, that is male and female gametophyte generation, and the gametophyte generation, that produces the gametes, and these gametes fuse to form zygote inside the seed, and this seed can then develop into a sporophyte. So this was about the life cycle of gymnosperms. Now moving on to angiosperms, flowering plants. Angiosperms are the flowering plants in which the seeds are embedded inside the flower or inside the fruits, if they have the ability to produce fruit. So before discussing the life cycle of angiosperms, we will be discussing these common concepts which you have already studied in the previous classes. Structure of flower. So a flower generally has both male and female parts within the same flower. So in the context of the reproduction, we will be discussing male and female parts of flowers. So you already have an idea that the male parts, the petals, sepals, and all the parts in a flower, they are forming a whorl, or they are forming a layer. The outer layer is green parts, they are the sepals. Collectively all the sepals are known as calyx. Then the next layer is petals. Petals are collectively known as corolla. Inner to the petals there is the male part which is known as stamen. Stamen has two parts, anther and filament. Innermost part is the female part. It has the following parts. The uppermost broader part is stigma. Then this narrow neck-like structure is style. Then this broader part is ovary. Inside the ovary there is ovule. And you already know that this ovary is the future fruit and this ovule is the future seed. Moving on, pollination. Pollination is the transfer of pollens from a male part of a flower to a female part of flower. So you all have an idea that pollination always occurs within the same species, because pollination cannot occur with any other species. So within the species there are two types of pollination: self-pollination and cross-pollination. Self-pollination is more basic type of pollination because it involves one flower, and this type of pollination occurs when pollen grains from the anther fall directly onto the stigma of the same flower. And self-pollination actually reduces genetic diversity. On the other hand, cross-pollination is a more complex type of pollination that involves transfer of pollen from the anther of one flower to the stigma of different flower. That different flower can be on the same plant or it can be on a different plant of the same species. So this is cross-pollination and it results in genetic diversity. Moving on. Now, sexual reproduction in angiosperms. I already mentioned that angiosperms are the flowering plants and they have the seeds. Seeds, unlike angiosperm, their seeds are enclosed inside the flower or inside the fruit. So a brief summary that the adult plant is just like the gymnosperm, it is again sporophyte. Phase is the main phase of the angiosperm. It is again dominant. As with the gymnosperms, angiosperms are also heterosporous. They can also produce two types of spores, microspore and megaspore, which will form an ovule that contains female gametophyte. Microspore is going to give rise to pollen grains, male gametophyte, and megaspore will form ovule that contains the female gametophyte. Right? Then inside anthers, microsporangia. Anther was the part of the stamen, male part. Male gametophytes divide by meiosis to generate haploid microspores. Microspores are again haploid, which in turn undergo mitosis and give rise to pollen grains. Each pollen grain contains two types of nuclei, two types of cells, generative cell and pollen tube cell. Generative cell produces two sperms and pollen tube cell has one nucleus that is going to form the pollen tube. We will discuss in the cycle. See, the ovule megasporangium protected by two layers of integuments and the ovary wall. Within each megasporangium a megasporocyte. Megasporangium is the part. Megasporocytes are the diploid spore mother cells, you can say. They are the mother cells and they are the diploid cells. They undergo meiosis to produce 4 megaspores, 3 small and 1 large. One large megaspore actually survives and it produces female gametophyte referred to as embryo sac. The female gametophyte is named as embryo sac. See, out of the megasporocytes are the cells, the spore mother cells, they give rise to 4 megaspores. Out of them, one is large and dominant, it is the surviving megaspore. It gives rise to female gametophyte, that is the embryo sac. Inside that megaspore divides 3 times to form 8 cells. In this picture you can see here, this is the embryo sac, and embryo sac contains the megaspore give rise to embryo sac, and inside the embryo sac the cell, the diploid, the haploid cell, it undergoes mitotic division 3 times. Mitosis. When mitosis occurs 3 times, how many cells are expected? In the first division 2 cells, in the second division 4 cells, in the third division 8 cells. Out of those 8 cells, only one of them is the egg. The mature embryo sac contains one egg, 2 synergid cells. They are present on either side of the egg, and then 3 antipodal cells. They are antipodal, means they are present on the opposite pole, or where the egg is present, they are opposite to the egg cell. And the 2 polar nuclei in a central cell, and in the center there are 2 polar nuclei within the same cell. So total there are 8 cells. So what happens due to these polar nuclei and this egg in angiosperms? There is a major characteristic of angiosperm is double fertilization. We will discuss. Here are the key points. See, microspores develop into pollen grains, which are the male gametophyte, while megaspore form an ovule that contain the female gametophyte. So microspore give rise to male gamete and megaspore give rise to female gametophyte, that is embryo sac. In the ovule, the megasporocyte undergo meiosis, generating 4 megaspores, 3 small and 1 large. Only one of that survives. This is a revision, that is known as embryo sac. When the pollen grain reaches the stigma, it extends its pollen tube to enter the ovule and deposit 2 sperms, because the pollen has 2 nuclei and 2 types of cells, pollen cell and generative cell. Generative cell produces 2 sperms. Pollen cell actually produces pollen tube. We will discuss in the diagram. The 2 available sperm cells allow double fertilization to occur, which results in diploid zygote. When egg is fertilized by the sperm, it is going to produce zygote, and when the central polar nuclei, they are going to be fertilized by another sperm. See, these are the central nuclei, n plus n, 2n. So when a sperm that is also haploid, it is added, it becomes 3n. So this is a triploid cell, which is the future endosperm. Endosperm is the major part of the cotyledons, which store food for the embryo in the seed. In the seed, you already know that there is a cotyledon, and inside the cotyledon there is embryo. The cotyledon has the endosperm, that is the endosperm. It stores food, and during seed germination, the seed cannot undergo photosynthesis, so it uses the store food in the endosperm in order to germinate. Here we go. This is the life cycle of angiosperm, starting from here. Here you can see that it is a flower, mature flower on the sporophyte plant. The plant is sporophyte, and on the sporophyte, this is the part which is flower. Flower contains both male and female parts. Let's discuss the male part. The anther has the microsporangium. Microsporangium contains microsporocytes. Sporangium is the structure. Sporocytes are the cells, that are the diploid cells. They are the mother cells. They undergo meiosis and they produce microspores. The microspore has 2 nuclei, generative and tube, 2 cells, generative cell and tube cell. So this tube cell is going to form, see, they are released in the form of pollens and they fall on the stigma, and here the pollen tube, it forms this long tube. Here you can see it grows until it reaches the ovule, and the generative cell produces 2 sperms. Here you can see 2 sperms are there. These are the sperm cells. 2 sperm cells are there. And on the other hand, we already discussed that ovule has the megasporangium diploid, and it produces megaspore. Only one out of the 4 megaspores, one is the surviving megaspore, and this produces the embryo sac, and inside the embryo sac This megaspore undergoes 3 mitotic division and it produces 8 cells, antipodal cells, 3 antipodal cells, 2 central cells, 2 synergids and 1 egg. So the two sperms, when they reach the ovule, one of them is going to fertilize the egg and it will produce zygote. See, this is the zygote. When the sperm cell fertilizes the egg, it produces diploid zygote, and when the another sperm cell fertilizes the central nuclei, it produces endosperm, which is triploid, and this ovule develops into a seed, and inside the seed embryo lies in somewhere inside the endosperm. It is surrounded by triploid endosperm, and this endosperm has the store food, and with the passage of time the seed coat develops and the seed is protected. When this seed is exposed to a suitable environment, it is going to germinate into a new plant. So here you also notice that there is alternation of generation between sporophyte generation and gametophyte generation. How? The sporophyte generation give rise to male and female gametophyte generation. Male and female gametophyte generation produces male and female gametes, and those gametes fuse to form zygote. This zygote develops into a new sporophyte plant. So this is alternation of generation. Here you can see the blue portion is diploid generation and the pink part is actually representing the diploid part. So this was all about the life cycle of angiosperms. Next SLO, seed dormancy. You must have heard about dormancy. Dormant, what does it mean? Dormant means inactive. Seed dormancy is the condition in which seed remains dormant or inactive. So the seed dormancy can be also defined as the state in which seeds are prevented from germinating, or a condition of rest which enables an embryo to survive the long periods of unfavorable environmental conditions such as water scarcity or low temperature. So this is seed dormancy. See why the seeds remain dormant or inactive? Because there is a specific season or specific time of seed germination where the seeds germinate into a plant. So before that the seeds remain inactive, and it is of great survival importance that it prevents seed germination in apparently favorable conditions. Apparently favorable condition means, for example, a seed might get a temporarily favorable condition. For example, in winters, if a seed is somewhere on the ground and if it is exposed to a hot water, suddenly a warm spell in winters, so if there wasn't this mechanism, due to that apparent favorable condition, it can germinate, and then later on it will face the harsh environmental condition and it will not be able to survive. So dormancy actually helps the embryo to survive and wait for the favorable or particular season. Germination or resumption of normal growth by a dormant embryo requires very precise combination of environmental condition. So after dormancy, that is the seed germination or breaking the seed dormancy, what does it requires? It requires certain combination of environmental condition. So here we go. Germination is the process of seeds developing into new plant. First, environmental conditions must trigger the seed to grow. Usually it is determined by how deep the seed is planted, water availability and temperature. So how it starts? When water is plentiful, the seed fills with water in a process called imbibition. Imbibition is through the process of osmosis. The seeds are going to take water, and the water activates special enzymes that begins the process of seed growth. And the seed, how it grows? First, it grows the root so that it can absorb the nutrients from the nutrients and water from the ground, and next the shoot is developed so that it can develop its leaves and it can be exposed to the sunlight, and then it will start photosynthesis to make its own food. So here is the anatomy of seed. You can see these are the cotyledons. Inside the cotyledon there is the embryo. Cotyledon is actually the endosperm which has store food. Here this is hilum, is the part of the seed where it is attached with the fruit. This is the seed coat which protects it, and in the embryo you can see epicotyle is the part of the embryo which is above the cotyledon, and hypocotyle is attached to the cotyledon. So first leaves of the embryo are known as plumule, and first root of the embryo they are known as radicle. Let's discuss the types of germination. Seed germination has two types: epigeal germination and hypogeal germination. Epigeal germination is a type of germination where the seed cotyledons are brought on the surface or above the soil. They remain above the ground. After germination, the cotyledons are above the ground. That's why they are known as epigeal. Epi means above. Epigeal germination. On the other hand, hypogeal means, hypo means below. So in hypogeal germination, the cotyledons remain below the soil. They remain underground. So what is the mechanism? The hypocotyle, remember, hypocotyle is the part which is attached to the cotyledon. Epicotyle is the part which is above the cotyledon. In this slide you can see, note it again. Epicotyle is the part of the embryo which is above the cotyledon, and hypocotyle is the part of the embryo that is directly attached to the cotyledon. So here what happens, in epigeal germination, hypocotyle, the part of the embryo that is directly attached to the cotyledon, it elongates and forms a hook. Thus, when it elongates and form a hook, it is directly attached to the cotyledon, so it is going to pull the cotyledons above the ground. Common example is bean, cotton, papaya, and you can take many other examples. On the other hand, what happens in the hypogeal germination, the epicotyle, the part of the embryo that is above the cotyledon, it is not directly attached to the cotyledon, it elongates and forms a hook. So when it elongates, it does not pulls the cotyledon, and the cotyledons remain underground. Common example is pea, wheat, maize, and rice. Here you can see epigeal germination. See, this is the cotyledon, this is the embryo. Embryo has this part which is not directly attached to the cotyledon, hypocotyle. When hypocotyle elongate, it will form a hook, and when it elongates, it pulls the cotyledon above the ground. After germination, the cotyledons they remain above the ground. On the other hand, in hypogeal germination, epicotyle actually elongates, and it is not directly connected to the cotyledon. So when it elongates, the cotyledons remain under the ground, and the plumule, the first leaves, they are turned off or they move, they remain on the ground, and the cotyledon is still there under the ground. Last law of today's lesson is vernalization. Vernalization, from Latin vernus, of the spring, means is related to spring. So is the induction of plants flowering process by exposure to prolonged cold of winter, or by an artificial equivalent. So you must have noticed that most of the seeds germinate in the spring. The reason is that those seeds, in order to germinate, the seeds have to be exposed to a prolonged cold condition, chilling environment, which is required for the germination. This is the reason that when fruits are ripened in the summer, the seeds cannot germinate, because for germination they need a very cold temperature and exposure to cold environment. So the low temperature stimulus is received by shoot apex or the embryo. So it's equally important for germinating seeds as well as for germination of leaves or for flowering of plants in spring. Most of the, for example, mature plant, take an example of mature apricot plant, so it is going to flower in the spring. So what is the reason? That the mature plant, the shoot apex of the mature plant and the embryo of a seed, they receive the temperature stimulus, but the leaves, unlike as you, if you remember in photoperiodism, it was due to the, in photoperiodism it was the stimulus was received by leaves, but here it is received by shoot apex or embryo. The duration of low temperature or chilling treatment requires varies from 4 days to 3 months. Temperature around 4 degrees is found to be more effective. So it can also be done artificially. Certain farmers can use this mechanism, giving exposure to very chilling environment before seed germination, can give better results. It stimulates the production of vanillin hormone, which is actually gibberellin. So gibberellin is the hormone which is responsible for seed germination. Here you can see in this slide, photoperiodism and vernalization serve to synchronize the reproduction behavior of plants with their environment, ensuring reproduction times of years. So we already discussed in photoperiodism that like long day plants and short day plants, certain long day plants, they have a specific season to like flowering is stimulated in specific seasons, and in short day plant it is again specific. So photoperiodism and vernalization are, they work in a synchronized manner. That is the reason that species of the same flowers, same plant species flower at the same time or in a particular season. So here you can see the effects of vernalization. This is a germinating seed, no chilling, plant it at ordinary temperature. Here the flowering is not stimulated. Here chilling for month at 5 degrees Celsius, plant it at ordinary temperature, then see the chilling has the effect and it has stimulated the flowering in this plant. So this was about vernalization. So this was all about today's lesson. Hope it was clear to all of you. Thank you so much.